Anti-neoplastic agents. Drugs used in treatment of neoplasm is known as anti-neoplastic. Neoplasm means medical term for cancer. You can also call them as anti-tumors or anti-cancers. Hmm? Shh. Anti-cancer or cytotoxic drugs. You can also call them in different names: anti-tumor, anti-cancer, or cytotoxic drugs. So the picture you can see it, and this is the normal cell. You can see normal cell. There is extra growth. You can see it here. You see there is extra growth on the surface of the normal cell. This extra growth, here also you can see it, an extra growth is there. This is known as cancer. It is like a crab. I think you people might have known many crab. It is an crab is a sea food. How the crab attaches the same way cancer cell attaches to our body. Cancer cell attaches to our body. Okay. So anti-neoplastic agents are the drugs used in treatment of neoplasm. Neoplasm means is a medical term for cancer. And they can also be called as anti-tumors, anti-cancer, and cytotoxic drugs. Now, see, according to American Cancer Society, according to American Cancer Society, cancer is a group of disease characterized by uncontrolled growth, and spread of abnormal cells. So what we are telling, I think you people are more familiar. Cancer is a group of diseases. It's a group of diseases characterized by uncontrolled growth and spread of abnormal cells. Spread. See, bone cells will be in the brain. Brain cells will be in the breast. The cells are unique everywhere, but they spread here and there. So spread of abnormal cells. Next, if the spread is not controlled, it can result into the death. It results in the death. And cancer can originate almost every part of our body. It can originate any part of our body. The most common site for women is breast. The most common site for men is prostate cancer, prostate gland cancer. We have discussed about these prostate cancer drugs last semester, I guess, last year. Please refer to the drugs whatever we discussed. And here we will discuss. Please go and check it again. Alpha 1 receptor agonist, that we have seen it. So it is for men now, it is benign prostatic hyperplasia. What happens is that it is not dangerous, but they block. Here it is the normal urinary tract. So this pink, blue color line, you see, it's a normal opening. So when you start maturating, what happens? Urine comes from the bladder. But because of prostate gland enlargement, what happens? Urine will not enter. It takes time for them to urinate. Or if it is, it is if it is more size, no, urine may not come. It is a benign cancer. We will learn what do you mean by benign cancer. This. Then women, no, it is the breast cancer, which is responsible for uh, highest prevalence in women is the breast cancer. Now, so cancer, what let us learn what do you mean by some terms. Cancer means unwanted growth. Unwanted growth. Now, it is obtained from the Greek word called carchirons, which mean crab. I was telling, no, this is called, this is the figure of crab. This is the figure of crab. You can figure it out, I think. Because the cancer cells adhere to any part of our body similar to crab. So they have given the name cancer. So crab, when it attaches to our body, how it attaches? Attaches? Similar way, cancer also attaches to our body. That's why they have given the word cancer. Then tumor. Tumor literally means swelling. Literally means swelling or mass of cells in the body which does not have any physiological function of the body, any physiological function. This is called as tumor. It is called as tumor. Next, neoplasm is a medical term for cancer. Medical term for cancer. Malignant. Malignant means cancer tending to become worse and cause the death is known as malignant. Is known as malignant. Then metastasis is a process by which malignant cells spread to another part to produce secondary tumor is known as metastasis. So these are the terms or definitions which come during study of cancer. So cancer means you learn, tumor means you learn, medical neoplasm you learn, malignant means worst conditions. Metastasis means breast cancer moving to brain, breast cancer attacking bone. So you can find breast cells in our bones, and you get the cancer there. So so this is known as metastasis. We will learn and which part will be responsible for secondary cancer. What is the mechanism? What is the mechanism for metastasis? This all we will learn here. Next. These are the properties or hallmarks of cancer. Properties of cancer are hallmarks of cancer. Self-sufficient in growth signals. What happens, no? If a cell want to grow, you will get a signal from other part, but cancer cell gets their own signals. So self-sufficient in growth signals. Insensitivity to anti-growth signals. They will not be responsive. They will not stop anti-growth, respond to the anti-growth signals. Evading or escaping to programmed cell death. They escape programmed cell death. Then limitless replicative potential. Limitless replicative potential. They become immortal. The cancer. See, developing blood cells. They generate new blood cells. We will learn why all this happen. What are the characteristics and everything. Then tissue invasion. that is nothing but metastasis, attacking another cell. Then tumor promoting inflammation. Even repeated inflammation will also cause tumors. Why? The reason we will learn. Gene instability and mutations. Huh? This term you are from the last 1.5 year we are hearing more. Same way like that. Gene instability and mutation. We will learn that also. No contact inhibition. Then deregulating cellular energetics. Deregulating cellular energetics. All these are the hallmarks or properties of cancer, or properties of cancer. So these are now self-sufficient in growth signals, uh, or abnormal signaling pathway is the first hallmark of a cancer. Now you can see it. Cancer cells do not need stimulation from external signal in the form of growth factors to multiply. What we told first itself during the studying of cancer, what we told, unwanted multiplication of cells, unwanted growth, we told. So to start growing, know what happened? We need some signals. Generally, know what happens? There are two types, paracrine and autocrine. Hormones are produced by one cell. They travel blood. They travel through blood and they act on different cells for growth. For growth. But cancer cell, now what happens is that they secrete their own hormones. What are that hormones or growth factors? We are telling this like this. Platelet derived growth factors. Transforming growth factor alpha, epidermal growth factor, vascular endothelial growth factor, basic fibrinoblast growth factor. These like this factors will be produced in our body more. Suppose if you take cancer patient plasma and normal uh, healthy person, I should not tell patient, no, healthy person plasma, and check, these factors will be more in cancer patient. And they also found that uh, a cancer cell can produce their own signals, their own signals. That means more hormones are produced. So what happens? More multiplication occurs at this place, at the place. So these are the growth factors. Suppose platelet derived growth factors, platelets will be generated more. So whatever the energy uh, uh, generated in our body will go to the places where multiplication are more, where division is more. So cancer cell will take all the energy which is produced in our body, which is produced in our body, and make the person very weak, very weak. So they produce their own receptors. See how they produce? They produce their own receptors by autophosphorylation. For example, you see ERB2 receptors, ERB's receptors, receptors in breast cancer is increased. ERB receptors in breast cancer is increased. Now these receptors is activated even at low concentration of growth factors. These ERB receptors will be more in breast cancer. So even for few growth factors, even at the low concentration of growth factors, what happened? These receptors will get stimulated. They'll get stimulated and starts multiplying. You see, all these are new terms. Hmm? You need to learn. Sometimes we don't get even full form of this. ERB receptors in breast cancer is increased. Now, next, sorry. Next is insensitivity to anti-growth signals. Insensitivity to anti-growth signals. First, what I told, producing their own signals, their own growth factors, their own growth factors. Now, what happens is increased growth of cell. Generally, what happens if there is increased growth of cell? There is increased inhibitory growth factor. Whenever whenever there is increase in growth of cell, what happens? Body will produce inhibitory factors, inhibitory growth factors. You're getting an inhibitory growth factors. What are that? We learned growth factors in previous. These are P53, P21, caspases, Bax factors. Some of them examples. P53, P21, caspases, Bax. These will get increased. And what happens in response to these growth factors? What happens? Cell growth and division is decreased. Is decreased. This case, if is when you are healthy. If you are healthy, then only this can happen. This can happen. Now, if the healthy cell is converted to cancer, what happens? The cell will not be effective to this growth inhibitory factors. So insensitivity to these anti-growth signals raises the risk of normal cell becoming cancerous by multiplication. These factors become inactive. They become inactive. So what happens is that decrease the activity of tumor suppressor gene. This is also all these are tumor suppressor genes are growth inhibitory factors. Growth inhibitory factors. So students, any doubt? Any doubt, please?
No.
No. You studied all this? Hello? Srinu, you studied this?
As in we've done a bit like overall, not completely in depth.
Okay. So this is the thank you. Insensitivity to anti-growth signals. Now, evading programmed cell death, which is known as apoptosis. Evading programmed cell death, which is called as apoptosis. Now, you can see it. Apoptosis is a form of programmed cell death. It's a programmed cell death due to aging, abnormality, if there is faulty cells. then uh, the process of apoptosis will get activated. Suppose normal RBC cell lifespan is 120 days. White blood cells, some of the white blood cells even have 1 hour. So after performing their function, they has to die. Then it is called as abnormal. Suppose if there is abnormal cell like cancer cell or faulty cell like cancer cell, then it also need to be killed. Hmm? It also need to be killed. This process is known as apoptosis. So now you see apoptosis is important in destroying the cells that escape from the normal tissue environment. I told you, no, suppose my brain cells cannot live in kidney, kidney cells cannot live in brain, or in what is that, breast cancer, breast cancer. So they need to live in their own environment, they need to live in their own environment. Suppose if they escape like that, then this apoptosis program will start. The button of apoptosis will be started and ruin that cell. But cancer cells, very tricky, very intelligent cells, you see, cancer cells are characteristically able to bypass this mechanism. Bypass this mechanism. So cancer cells, even though become grossly abnormal, we learned this is a simple definition. Abnormal cells, they do not apoptosis. They do not apoptose. Uh, Cancer cell do this by altering the mechanism. How does they alter? How does it happens? By altering the mechanism of apoptosis, by improper functioning of these factors. These factors, previously inhibitory factors we told, no, or tumor necrosis factors we told, that is only tumor necrosis factor, caspases, P53. When I was a student, I learned only P53. In that Lippincott textbook, latest they told in that book P53. Now they're telling P27, P21 gene, then Bax proteins. So they alter these uh, factors in the body. So let us learn how does this happen in a diagrammatic representation. Here. It can occur by eccentric pathway, like blood coagulation process, eccentric pathway, and even by intrinsic pathway inside the cell. Eccentric means outside the cell. T lymphocyte is the one which plays very important role in our body. This also last time when we given the assignment, you might have come across this. Even in coronavirus, they used to tell T lymphocyte is the one which plays very important role. This, no, it will kill every day at least one cancer cell. So cell division is a normal process. So there may be faulty generation of cells. So who will do the policing work or vigilance work? This is the T lymphocyte which does vigilance work. They patrol our throughout our body and kill all the unwanted cells. So T lymphocytes, when enter into cell, it becomes gramzyme, granzyme. Then it enters into the mitochondria inside again. Then it becomes, you see, different parts. It becomes cytochrome C, then apoptosome, then caspases 9, procaspases 9 caspases, which causes death, which causes the cell death. Then low levels of growth factor. Whenever low levels of growth factors are present, growth factors like tyrosine kinase receptors. When there is low levels of growth factors, tyrosine kinase receptors, growth factors are attached to these receptors. What happens? Signal transduction will be very weak. Then this will give signal to the mitochondria. Mitochondria will produce cytochrome C again. Cytochrome C. Apoptosome. Apoptosome is formed. Then again procasposis. Then casposis. Then it kills the cell. Then one more method is death activated protein. Suppose if it gets activated by tumor necrosis factor, signal transduction will occur. It directly activates caspases 8. Aspaces 8, which can kill. Or it can indirectly produce BID factor, which again activate mitochondria to release cytochrome C. Then again usual process. This is all from the outside the cell. Suppose if it is from the inside the cell, then it is called as intrinsic pathway from the nucleus. DNA damage, because DNA is produced. You might have studied even in our types of receptors. Types of receptors. That is nuclear receptors. So nucleus will produce DNA. Whenever there is faulty DNA, what happens? P53 gene, P53 protein is produced. Then it produces bags. Then again it activates mitochondria. You get apoptosis. You get apoptosis. Sorry, children. This isn't. This is called as programmed cell death. This is how it occurs. This is how it occurs. But cancer cell escapes by activating this BCL2. BCL2. So it blocks this process, apoptosis process. Lot of research is going on to find to stop this process. To stop this process. But what is the problem is that it needs to selectively go and block the cancer cell only. It needs not to block the process which is occurring usually in our body. Usually in our body, because many cells die every minute in our body. It's a usual process. Cell What is that? GIT lining, hair follicle, skin, new cells will be generated. So this is an name. This is also one of the uh, important they found it. Limitless replicative potential or cell division. Limitless, it becomes limitless. So what is the reason for that? You see how the research they have done. This is chromosome of a person. They found the pink portion. The name of this portion is called as telomere. Telomere. So what is the function of this telomere is that it helps in replication process. We told that DNA is converted to DNA. Many times we learned this. DNA is converted to DNA. This is called as replication process. So how many times DNA will converted to DNA? This telomere plays very important role during this process. Telomere shortens after multiple replication, something around 60 to 70 times. What happened? It can multiply, then it will shorten. So when it shortens, what happens is that then telomere at senescence will come. That means there is no chance of now replication. There is no chance of replication. But cancer cells have a character upregulating telomerase. The enzyme telomerase. What is this enzyme? What it does? The enzyme that maintains telomeres. So usually it is causing 70 replications or 70 divisions. After that, it is unable to divide. It is unable to divide. But because of this telomerase enzyme, what happened? Then this one will become again to this. So again replication, again 70 times. Again this enzyme gets activated 70 times. So DNA will be continuously replicating. So this they found. This is immortality. This is the immortality what we have discussed. Limitless replication property. Are you getting, children? Any doubt here? So cell can now divide indefinitely without entering senescence. Senescence is the property where DNA will not, a chromosome cannot divide again. Cannot divide again. So this is this is the property. So children, any doubt here?
No doubt.
So this is they are trying to check by the help of this. They want to make a human to be immortal, but I don't think they will get successful. They won't get successful in this, making humans uh, immortality. Now, angiogenesis. You see, angiogenesis. What is angiogenesis? Generation of new blood vessels. Angio means blood. Genesis means development. Is the process by which new blood vessels are formed. So now cancer cell appears to be able to kickstart, is able to kickstart the process. Which process? Angiogenesis. So you can see it. Suppose if the small cancer cell has started forming here, what happened automatically? Blood vessels. You see, like all the capillaries, new capillaries will be formed within uh, within months. Full complete picture it will come out. New blood vessel. What is difference between these blood vessels or these capillaries or normal capillaries is that these capillaries are very leaky, very leaky. They you can find using of exudates from the blood vessels. They're abnormal grown. That's why they're very leaky. So why this is required? They want to supply food, oxygen, because complete it is metabolically active. It is metabolically active. Even you can see it. And at this age, you people eat more than your parents. If you want, you can check it. Why? Because you are metabolically active. You are in phase of growing. After 40, suppose like me, 40 club, then what happens? It becomes less. This is it. So all the oxygen, nutrients will be providing by these blood vessels. What are the factors? You see, vascular endothelial, basic fibroblast growth factor. You need to write like this only, small d. Basic fibroblast growth factor. You see, this is it. Let us see how does it happens in next slide. Now you can see it. You see, rapid growth in there is rapid growth in cancer cell, then increased demand oxygen and food material. When there is increased demand, what happen? Hypoxic inducible factors. Automatically oxygen will not be enough, and food also will not enough. So there is release of some factors. So they have given that hypoxic inducible factors, HIF. Examples are VGF, VEGF, sorry, BFGF. I told you, no, vascular endothelial growth factor, basic fibrino uh, factors. These factors, what happened? You see, these factors are increased. These factors interact with the receptors on endothelial cells. Blood vessels are nothing but endothelial cells. You can see it. Definition of blood vessel, if you see, it is endothelium. Endothelial cells are made up of nearby blood vessel and stimulate these cells to divide. So endoepithelial cells will be divided, leading to branching. You observe the previous figure. So small networks. It is forming. It is forming networks. So branching, extension of capillaries is called as angiogenesis. Angiogenesis. The newly formed blood vessels are diagnosed in structure and dilated, leaky. They are very leaky, disorganized. Sorry, the newly formed blood vessels are disorganized in structure, dilated, and leaky. They also produce integrins. They also produce integrins absent in normal cells, which are absent in normal cells. What is the what is the function of these? These integrins block apoptosis. Some research has been done. A lot of research has been done. Some of the drugs are successful with this uh, with this factor, with this to block this hallmark. So, for example, thalidomide, which was used in morning sickness, we learned many times that they have withdrawn from the market. Now they started again this thalido, thalidomide. So this thalidomide, then endostatin, you see, stop, endostatin, angiostatin, cambristatin are some of anti-angiogenic drugs. Some progress has come and some drugs have been released here. That's why we specified here. So this is one more factor, angiogenesis, hallmark. Then tissue invasion and metastasis. So the normal cell which has to be present in its environment will escape from its place. It escapes from its place and it escapes from its environment. You see, cancer cell can break away from their site or organ to invade the surrounding tissues and spread to distant body. For example, you see, normal cell, no, what happens? Like this is one cell, this is another cell. Now, in between, you see, like a thread, there is some substance which is called as cadarin. Cadarin. This cadarin links the two cells. Now, this portion will get deteriorated. Then what happens? You see, this has been detached. This has been detached. Then collagen becomes weak. Collagen becomes weak. So they come closer. They come to closer to the blood vessel. Then type 4 collagenase enzymes comes there and it escapes from that environment. Migration. You can see it here, loosening of intracellular junction by removing of cadarin, then attachment to the basement. Then what is that? Collagenase enzyme will cause this collagen to become weak, so this cell can move away from its environment. So for example, you consider this is a bone cancer. Now this bone cancer can go to the liver. You can find bone cells in the liver cell like this. This is called as metastasis. So let me have little water. This cough is not due to COVID. This is due to my gastritis. I talked with the doctor. He told if you this ulcer and gastritis are reduced, automatically your cough will suppress. If I may come to the university, they may send me out. Thank to God, I'm active now and taking class. I was totally sick in those days. Some people asked me to make a video. I became almost unconscious. Thank you. Please sign. There are 52 students in the meeting. Only 41 people have written their names in the chat. Please do sign. So this is it. Then here also lot of research has been done. This is avoiding immune system, avoiding immune destruction. Cancer cell will escape immune cells. Immune cells. So you see here, majority of small tumors are destroyed by immune system. Example by T lymphocytes, natural killer cells. Natural killer cells. But large tumors avoid detection by immune system by masking antigenic property. Suppose a foreign substance arises in our body. When it enters into our body, what happens? Antigenic property. Foreign substance is nothing but antigen. So you get antigenic property. In response to that, antibodies are produced. But these cancer cells will not produce that antigenic property. How? By inhibition of stimulatory signals of immunity. They will not activate immune system. Decrease the function of antigen presenting cell. APC, we call it as APC, antigen presenting cell. So they decrease this functional. Decrease the function of interferon. Decrease the function of interferon. Decrease the function of tumor necrosis factor. Reduce the number of natural killer cells, T lymphocytes, and thymus cells. All this, because of all this, immunity is decreased, or cancer cell cannot be detected by immune system. Immune system. So, a lot of research has been done. There are some drugs are also into the market. What I will do is that I will give some links. You can read them if you are really interested in this. Now, suppose you see greater incidence of cancer, is observed in immunosuppressed individuals. HIV, for example, HIV, autoimmune diseases, patients with consuming immunosuppressant drugs. Why? Because the cancer cells are escaping from immunity in healthy person. If you are taking these drugs, or if you are having HIV or autoimmune diseases, or if you are taking these drugs, immunosuppressant drugs, then still immunity is decreased. You might have studied in black fungus because of overuse of dexamethasone. What happened? Immunity has been reduced. So that's why black fungus has caused the death for the COVID patient. We learned last time. Some of you people have presented the term paper presentations. So that is also one example. Why immunity plays. Immunity, HIV, acquired immunodeficiency syndrome. When immunity is decreased, automatically HIV infection gets more. So in HIV cancer, cancers are more predominant. So let me go fast little. Then finished. Next, tumor promoting inflammations. Tumors are promoted even by inflammation. They check the normal cell and tumor cell. Then they found that the COX-2 enzyme is expressed more in tumor cells. We learned that COX-1 is pathological or housekeeping enzyme, Whereas COX-1 is physiological, COX-2 is pathological pillar. So COX-2 is more expressed in tumor cells. So they concluded that in chronic inflammations, that is autoimmune diseases, bacterial and viral parasites, can cause normal cell to develop pro-neoplastic mutations and resist to neoplasis. Oh my God. Resist to apoptosis. Apoptosis. So, and one more thing is COX-2 is overexpressed in many tumor cells. Why? Because you see inflammation recirculate leukocytes such as neutrophils, macrophages, which releases cytokines, growth factors, which aid nourishing the tumor. So because of this, they are telling even inflammation is one of the characteristics for tumor promoting inflammation. Then last, then gene instability. Gene instability, we will learn this in coming slide. Tumor cells continuously evolve by mutation of genes like p53, PRB, growth factor genes, etc. They do some mutations, they change their activity. Contact inhibition, you see. So now in our university, suppose in class, now what happened? For one table you sit one person. Suppose if we have students and a dean meeting, what happened? All the students of 280 of pharmacy students will accumulate in one class. So on a table, how many people can sit? Maximum 3 people. Then your bodies will tell, we are contacting more, no further. Yes or no? Or 2 people can sit, okay, with 3 people with more difficulty. 4, no. But in cancer cell, what happened? How many cells they grow? There won't be inhibition. There won't be any problem. So when clumps of cell growth, then signal arises to stop the cell division. That is overcrowded and stop division. But in cancer cell, this will not happen. Let us see some diagram. You will better understand little more. So sorry, this is using normal cells show contact inhibition. You see, there is cell to cell some gap. But whereas cancer cells lack contact inhibition. You see, like best friends. In a room, 10 people also they can sleep like that. They are there. Then you can see normal cell grow in layer form it grows. So you can see the laterally how the growth is. In cancer cell, you can see lumps are formed, clumps, see growth. That's what we are telling. No, cancer is nothing but a crab. Crab attaches to our body, how it looks like that. There is clumps, there is hump like is formed. These are the properties of cancer cell. So what happens? They keep growing. This is one cell multiplying to 2, 2 will come to 4, 4 8, 8 16, 16 32. Multiplication, growing. Because there is no contact inhibition, no contact inhibition. Now last one. Don't go away, we'll finish this topic. Anyhow, afternoon also you don't have class. What is time? 4. Oh, it's only 1 hour. Another 40 minutes is there for me. Huh? Now, you see here, normal cell in presence of oxygen, glucose, it uses glucose, uh, and you get pyruvate. You get pyruvate. During this process, you get 36 molecules of ATP molecules. 36 molecules of ATP. And pyruvate is least converted to lactate. Is least converted to lactate. Now, in absence of oxygen, what happened? Glucose is converted to pyruvate and pyruvate is converted to lactate. Pyruvate is converted to lactate. How many molecules of ATPs you get in absence of oxygen? You are getting only 2 moles of ATP in absence of oxygen. Now, this is for normal cell. For proliferating cell or a cancer cell, whether it is presence of oxygen or absence of oxygen, glucose is converted to pyruvate. Pyruvate, only 5% of pyruvate enters to the cell, remaining is converted to lactate. And you will get only, see, 4 moles of ATP, 4 moles of ATP. 4 moles of ATP. Remaining is converted 85%. You can see it, 85% is converted to lactate. That's why the cell pH of cancer cell is acidic in nature. They tell even now acidic foods will increase cancer. Acidic food will increase all your colas. What is that? Fermented food will increase the pH. Sorry, will decrease the pH. That is acidic in nature. Acidic in nature. So this is how this, because of this, what happened? We get weak, and all the energy produced, that whatever the 4 ATP molecules produced, is taken by the cancer cell. Is taken by the cancer cell. So this is an. So any doubt here?
No.
No. Very good. So let me take further step, or you can check it, recollect yourself. I will be running the slides again. By the time I can take little time relaxation. So at any point of movement, if you have doubt, you can ask. These are the properties we learned. This is the cancer diagram we learned. We showed last 8 slides. We improved little by adding pictures. Last year also I kept, for the previous, no, I didn't do these pictures and everything. Then this diagram was there, this also was there, then these terms. See, now here you see breast cancer cell move to bone. Anyhow, we have one slide. uh, Brain tumor. So breast cancer can cause bone cancer and brain cancer easily. Mm, Anyhow, breast cancer, please, that's what I'm telling you. Don't drink cola once in a month, like that. Some people, I find, no, Instead of drinking water, they're drinking Coca-Cola or Sprite. It's very dangerous. You are troubling yourself. No one in the world huh, will will take carbon dioxide to the body. Everyone knows that carbon dioxide is bad for health, but many people are taking it. So please avoid that. And most of the times, at least drink 3 liters for, it's a general suggestion, 3 liters, usually 3 to 5 5 liters, 4 to 5 liters is the hmm, correct consumption of water. At otherwise, you drink at least 3. Morning 1 liter. By going to home, you drink 1 liter. After going to home, before going to sleep, you drink at least 1 liter of water. Water no cleanses our body. Suppose if you take a shower, how happily you will feel. Same thing will happen when you take inside. Your inner environment will be very healthy. Give chance to the body. It will automatically improve life. And most of the times I can see it, and you are eating more processed food. You means all of our in the world it is happening. And uh, we all, let me to make the sentence, we all eating processed food, that also bread. It is white flour, it doesn't have any proteins. That also avoid. That also avoid. Once in a month or with family you can eat. I'm also telling that. I do order Domino's pizza. I do eat once in a month or like that. Hmm? But do not order every day, please. Please take care of you. These are general instructions for you. Now, this is immune system escaping, tumor promoting. This, this. So now you see cancer types in US. Why we are telling US is that the data is completely available. It is almost same even to the world. You see, uh, in male, no, the more predominant cancer, I told you in the first slide itself, prostate cancer, women, breast cancer. Then both lung cancer, colon cancer, these are becoming predominant. Colon. Lungs means pollution. We can't do that. Then urinary bladder, uterine, then skin cancer, ovary cancer. So these are the cancer, different cancer in men and women. But death due to cancers, estimated death is more with lung cancer in both sexes, in either sex. Next is prostate cancer, next is breast cancer. Next again colon cancer. Colon cancer can be controlled if you take good habit food. Hmm? Make at least 30% uh, healthy food. That means vegetables. Whatever the available fruits in the local market you eat, clean them well and eat it. Hmm? So this is an. So these are different. Then as age increases, hmm, the number of cancer cells, cancer patient increases. You see, 40 people. When you are age between 40, it may be near to 100. But when it comes to between 50 to 54, 500 patients. So as age increases, what happens? The predominance of cancer is increasing. Is increasing. Mm. Classification of cancer. Two types, just simple learning, we'll run fast. Benign cancer, malignant cancer. Benign cancer is non-invasive. I know you people know. Malignant cancer is invasive metastatic. There is well-defined borders, irregular borders, because it is escaping from its environment. It is like migration now, huh? Because of globalization. Everyone is moving, huh? Like that, malignant, invasive. Then well-defined. Benign tumor is well-defined. You can remove it. Benign cancer, if it is there, no, you can completely remove by surgery. So regular nuclei, rare mitosis. Rarely it mitosis. Then poor differentiation, irregular and large nuclei, more frequently and abnormal mitosis, escaping to another environment. Among all these, both common factors is that abnormal growth and contact inhibition. Both. For the common feature of both of these is these properties: swelling, abnormal growth, and contact inhibition. Contact inhibition. Next, benign tumor. We learnt about that, well defined. What is the we told this is problem, not a problematic, but whenever benign tumor is coming across a blood vessel or nerve, suppose you consider this is a colon. Now if there is growth here, what happens? Colon will be blocked. Colon, large intestine, it will be blocked. For example, you consider this is a blood vessel. If there is growth, what happens is that it stops the blood supply to that. Consider if this is a nerve. If there is growth here, what happens? Nerve supply to the organ endings will be stopped. That is problematic. Until these places, that is the growth of the cell near blood vessel or nerve endings or GIT tract, there is no issue with colon. What is it? Benign tumor. Benign tumor. Now you can see it, and benign tumor, they are very happily living. You see how big it is, but he survives. You see, in the mouth, hyperplasia. We have seen this is a side effect for phenytoin. Here you see, only problem is that he can't chew. Now here you see, he can't see. He can't see that because of benign cancer. Entire face has been closed. Next, this woman, you see how big it is. So this is the problem with the benign cancer. Okay. Now, malignant cancer, we learned metastasis and all this. Then, preferential metastatic process. Where do we get that? So, breast cancer, breast adenoma carcinoma. It moves to bones, brain, adrenal gland. Adenoma carcinoma to bone. Lung, small cell cancer, carcinoma. It moves to second. All these are secondary cancers. The sites, these are primary tumors. These are secondary tumors. The frequently moving. Suppose if you get breast cancer, there is chance of either bone cancer or brain cancer or adrenal cancer. After research, this table has been presented to us. Suppose if you have lung cancer, bone, brain, liver. If you have thyroid adenocarcinoma, bone. Kidney, clear cell carcinoma, bone, liver, thyroid. Testis carcinoma, liver. Bladder carcinoma, brain. So these are the preferential metastatic sites. This. So this is now Cancer cell classification by structure. By structure. You see, carcinoma. Carcinoma means 80% of cancers belongs to carcinomas. Cancer in epithelial cells is called as carcinoma. Mainly skin cancer, liver cancer, colon cancer, prostate cancer, lung cancer, breast cancer falls under all these carcinoma. Now, sarcoma, it constitutes 2%. Cancers in connective tissues or non-epithelial cells are known as sarcomas, like bone cancer, muscle cancer, fatty tissue, like osteosarcoma, gum cancer. Then leukemia, leukemia, it is liquid cancer or blood cancer. A number of white blood cells will be formed over. Then lymphomas develop in glands or nodes are known as lymph glands. Then both what is that, leukemias and lymphomas constitute 15%. 15%. So these are different classification based upon the structure of cancer. Then how does these normal cell, healthy cells are converting into cancer cell? This is the mechanism, molecular mechanism of cancer or oncogenes, oncogene. We will learn what do you mean by oncogene and normal healthy cell or normal cell is converting into cancer. Why? Because of irradiation, viruses, and food. A mutation in normal, there is mutation. All these where little by little we will see and will learn. Now, see, what are mutations? So there is change in nucleotide sequence of DNA. DNA, G will bind to cytosine. We learned in cancer now, but they change this. Then it is called as mutation. Change in nucleotide sequence of DNA may occur in somatic cells. If the change occur in somatic cells, then what happens is you only suffer. You don't, your offsprings will not suffer. Suppose if these mutations occurs in gametes, like egg or sperm, what happen? Offsprings will suffer. So my parents have hemophilia, for example. If parents suffer, children also suffer. It is that. Mutations occur regularly. For example, you see, mutations occur regularly. Our body will repair them regularly. There is vigilance, programmed vigilance cells, which will take care of these mutations. But sometimes it becomes abnormal. We will study that. Almost all mutations are natural. Chemical and UV radiation causes mutations. Mutations may be repaired by enzymes. Let us see that. Don't leave. Another 20 minutes. We have time. Now, what are the types? You see, point mutation, chromosomal mutations. Here, deletion, translocation, insertion. When you learn, no, you will understand. I'll go very fast. Don't worry. Point mutation. Now, you see, this is the when person is exposed to UV light, you will get such type of cancer, skin cancer. This is skin cancer, which is known as xeroderma pigmentosum. Xeroderma pigmentosum. What happens here? You see, like this on the face, or whenever they exposed to UV light, some you can find some of your parents getting like this. What happens, you see? For example, hmm, thiamine, they combine with UV light. hmm, in presence of UV light, they form a adduct substance. Adduct substance. Actually, they are separated. Now they became combined together. They combine substance. This is called as adduct substance. Then thiamine cytosine. Hmm? Thiamine cytosine. They form in presence of UV light. What happened? They also form adduct substance. These structures are cyclobutane structure. What is this ring? Cyclobutane structure. Hmm? These uh, these changes occur even with all normal people. Even me, if I don't have any skin cancer, if I go to UV light in midday, what happens is that I get it occurs in our body. But what happens when you come to shade, or when you come uh, to shade after when you come away from the UV light, there is proteolyase enzyme in our body. Proteolyase enzyme in our body. You can see it, and this reaction is reversible. This reaction is reversible. Proteolyase. What does this proteolyase does? It breaks that cyclobutane structure. Whenever you come to shade, it occurs. Even with the thiamine and cytosine, it also occurs in the same way. With the proteolyase enzyme, a proteonut, photolyase. With the photolyase enzyme, what happened? This cyclobutane structure reverted again back to this thiamine and cytosine. So if this proteolyase photolyase proper enzyme activity is good, you don't get this cancer. You don't get this cancer. If there is abnormality, exposure to UV continuously, for example, workers, hmm, what is that? Mansions, they continuously work in sunlight. For them, cancer. Any abnormality, then you get this this cancer. What is that? I'm not getting the name. Xeroderma pigmentosa. Xeroderma pigmentosa. So this is it. Hmm? What is UV light radiation range? We know now 200 to 400. So only thiamine and cytosine can undergo this type of reaction. It cannot activate adenine, guanine to undergo this process. They cannot. UV light doesn't have sufficient energy to activate purines. To activate purines. So this is one change which causes cancer to our body. Then chromosome mutations. See, chromosome mutations. What happened? Changing the structure or loss or gain. Let us see one by one. Then deletion. You get retinoblastoma. Say deletion. Sometimes there is removal of gene. Huh? In chromosome 13. In chromosome 13, what they found is that in the patient who is having retinoblastoma, when they checked at chromosomal level, they found some changes in chromosome 13. which is unusual when you compare to person. Healthy person. So what they found is that there is gene in chromosome 30 that is deleted. This portion, I didn't get the name of that gene. You see, due to breakage, a piece of chromosome is lost. In which chromosome? Chromosome 30. So because of that, you are getting retinoblastoma, cancer, tumor in the eye. This is because of deletion. Now, translocation, escaping one gene to another gene, is known as translocation. Example is formation of Burkitt's lymphoma. Now, you see, chromosome, this translocation is occurring between chromosome 8 and chromosome 14. There is, you see, this red, this portion, this portion is called as Mickey gene, MYC, Mickey gene. In chromosome 14, this is called as VH chromosome. VH chromosome. Now, there is translocation of Mickey gene from 8th chromosome to 14th chromosome. And VH chromosome will shift from 14th to 8th chromosome. Translocation it is occurring. So these changes you are observing in these Burkitt's lymphoma patients. Burkitt's lymphoma patients. This is it. Then mutation by insertion. Viral gene, you can see it. Virus gene enters into normal gene, then you get viral proteins and viral proteins. Then last topic, finished done. Only 3-4 slides. Please stay. Again, Y to N. Proto-oncogenes. What do you mean by proto-oncogenes? Proto-oncogenes. Proto-oncogenes are genes which are normally coded for proteins. Proto. You see here, proto-oncogenes. These are nothing but healthy genes. Healthy genes. What they do? These genes will code for healthy proteins. Healthy proteins involved in cell division and differentiation. So when there is proto-oncogene, what happened? Normal growth, normal regulated growth you observe. But whenever you see the gain of mutation, when you see proto-oncogene getting mutation, what happens? It is converted to oncogene. Proto will go, it will become oncogene, and that leads to cancer. That leads to oncogene. So once if oncogene is formed, then it is very difficult to control. It is very difficult to control. So proto-oncogenes are very good. They control us. Whatever we studied, normal cell, they behave like that. Now, what happens if it gets to proto-oncogene? The defective version. What do you mean by oncogenes now? Onco means cancer. You can see it in nowadays in hospital, oncology department. That means onco means cancer. This is also one more term. Huh? The defective. So oncogene means what? Defective version of proto-oncogene is known as oncogene. So proto-oncogene is converted to oncogene by the help of mutation, whatever we have seen, no, by that. So then what happens? Uncontrolled. So it is like a next diagram. You can see it. See, normal gene, regulated cell growth. It is like a with a good control break. If you have, uh, if your escalator and brakes are working well, then they are called as proto-oncogenes. Suppose if there is mutation, then what happens? Your brake is not working properly and your accelerator also not working properly. What happens? You get accidents. You get accident. So what are the examples of oncogenes? Now, you see, it's simple. And proto-oncogene means, for example, for proto-oncogene, one example is RAS protein. RAS protein. If it gets mutated, then it is called as oncogene. That is nothing but mutated RAS. What happens here? If it is mutated, always active, always stimulating, always proliferating. Proliferating in nature. So this is. So this all that only you can read by yourself. There is one more. Translocation. Hmm. Rearrangement or translocation. This is here between chromosome 9 and chromosome 22. One more example. See here, in chromosome 9 there is ABL gene, ABL, this red one. The ABL gene, BCR ABL gene. Then here there is BCR gene in 22 and there is extra pink gene here. What happens is that this ABL gene will shift on to 22 and the pink will shift on to the chromosome 9. That causing chromosome 9 still longer in size. The ABL gene on chromosome 9 has been translocated onto chromosome 22, right next to BCR gene. BCR gene. This makes a fusion of two genes that would not normally be together. This is called as BCR ABL gene. Previously, this is BCR. This is ABL. Now they are mixed together. So it is called as BCR ABL fusion gene. In where? In 22, chromosome 22. So this type of chromosome you find mainly in leukemia patients. In leukemia patients. Another name for BCR ABL fusion gene is known as Philadelphia chromosome. In US, there is one place called as Philadelphia. In that place, this gene in chromosome 10, 22 is found. The people who are living in those in that place in Philadelphia is having VCR-able gene. So they have given on the credit of that city, Philadelphia chromosome. This is also one of the example for translocation. Then results of gene, I told you, replication more, growth factors more, continuously stimulating this. So see, platelet derived growth factor. It will be normal. It will be proto-oncogene. If it gets mutated, it becomes oncogene. It gets oncogene. All these RAS protein, RAF protein, SARC protein, uh, uh, EGF, all these can convert it to oncogenes. I told you first here itself example. What is difference between oncogene and this? Proto-oncogene means RAS. Oncogene means mutated RAS, like that. Then even you can see it in some of the causes for cancer: chemical, radiation, virus. Normal cell will become damaged by damaging DNA. Then mutation occurs. Then activation of growth, inactivation of tumor suppressed gene, which leads to malignant cancer. So this is one more uh, carcinogenesis method. All this in one. Then you can see it. Read this. What is the important thing is that uh, they are successful. They are successful in controlling the cancer in the lab, but not in living cell. It's not in the living cell. So what is the statement you see? I got very good statement. It is easy to kill cancer, but the challenge is keeping the patients alive at the same time. So patient has to be mentally, physically very strong, especially family. Family has to support the cancer patient. They should not deprive him or her by giving separate room, not talking. You need to go to the patient, talk, find time, spend some quality time. You can see some of the diagrams. You see here, this woman. You see, Hmm. Hmm. You observe this picture. Still, she is loving her husband. Hmm? The face is the index of mind. So that face is there. Beauty also face plays very important role for the beauty. But still she is supporting her husband. So it has to occur in reverse also. It has to occur in reverse. So this you keep, if anyone is suffering, no, you support your family, hmm, then God will help her. So this is my lecture class. Any doubt, please?
No doubt.
Okay, then bye.